Now it's time to look into this business. Meiosis is a really important part of sexual reproduction. Remember, it's a type of cell division that only goes on with the sex cells. It is super important to understand that meiosis starts with one cell, then creates 4 daughter cells that are not identical to the original cell. Sounds juicy.
Before the process of meiosis starts, the cell is just chilling out like a regular cell. It's 2N, meaning it has 2 sets of chromosomes, one from mom and one from dad, just like every other cell. But guess what? The reproductive cells, or sex cells, are the only cells in the body that need to go down to just one set, or a single N. We'll talk about why this is later. But for now, remember that cells with 2 sets, or 2N, are called diploid, and cells with 1 set, or N, are called haploid. Think half for haploid. Meiosis is about going from a diploid cell to a haploid cell.
Let's get our bearings. We said that meiosis occurs in 2 rounds, which each contains 4 phases. The first round, meiosis I, has prophase I, metaphase I, anaphase I, and telophase I. Meiosis II, the second round, has prophase II, metaphase II, anaphase II, and telophase II. What a trip! Let's check out a little home video of this process.
Before we watch, let's just take note of one thing that's going on here. We know that a human cell has 46 chromosomes. That's 2 sets of 23 chromosomes each, right? Well, that's sort of a lot to draw for our models, like putting 70 candles on your grandma's birthday cake. So we're just going to use 6 chromosomes with 2 sets of 3 to represent a full cell. The set from mom is pink, and the set from dad is blue. Okay, hit it.
Just like in mitosis, before meiosis even starts, there is a period called interphase, when the chromosomes get duplicated into 2 sister chromatids, which are like Siamese twins attached at the hip. Now pay attention to this. Although the DNA has been doubled, the chromosome count stays the same. That's because 2 chromatids are still only considered 1 chromosome. I repeat, even though the amount of DNA has doubled, the cell is still considered to be 2N, not 4N like in mitosis.
2 chromatids stuck together are still only considered 1 chromosome. It's wrong. It's not fair. But you may as well accept it. They must be separated to count as 2 chromosomes.
The first phase of meiosis I is called prophase I. At this point, the cell is 2N, diploid, with 2 identical chromatids per chromosome. Now check this out. In meiosis, the chromosomes don't just come together, they also line up in pairs called homologous pairs, or homologous chromosomes.
Let's make sure we understand what homologous chromosomes are. It's really simple. Cells have 2 sets of chromosomes, one from mom and one from dad, right? So, the set from your dad has a chromosome that contains a gene for eye color, and so does the set from your mom, right? Well, these 2 come together as a pair so they can negotiate which eye color their child will have. The 2 chromosomes with the genes instructing for hair color come together, and for the length of your toes, too, and so on. That's what we mean by homologous chromosomes.
We're going to represent homologous chromosomes here by size. The big pink chromosome from your mom lines up with the big blue chromosome from your dad, and the small pink with the small blue.
The pair of joined homologous chromosomes is called a tetrad, which means a group of 4.
Don't forget that each chromosome has just been duplicated and contains 2 chromatids. The 2 from your mom are identical, and so they're both arguing for blue eye color, while the 2 from your dad are identical, and they're both arguing for brown eye color. That's 4 chromatids for each tetrad, and they're all talking about the same feature, in this case, eye color.
The next thing that happens in prophase I is really important, so listen up. It's called crossing over. Let's see that video again.
When our homologous chromosomes are in tetrad formation, they do more than just line up. See how some of them get crisscrossed and the colors get a little mix-matched? That's called crossing over. One of your mom's chromatids is like, okay, I'll consider brown eye color. And one of your dad's is like, fine, I'll consider blue. Gosh. And so they both mix their ideas, aka genetic instructions, together a bit. This is how you end up carrying different combinations of genetic information from your parents.
At the end of prophase 1, the nuclear envelope and the nucleoli begin to disappear, similar to mitosis. Now, in metaphase 1, they're all gone, and the tie-dyed tetrads line up along the equator of the cell, getting ready for some action.
In anaphase 1, the homologous chromosomes separate, guided apart by those jellyfish-like spindle fibers. The chromosomes have had their conversations, and now they go their separate ways. But remember, they still have their chromatids, which have not been separated yet. But guess what? The chromatids are not all identical anymore. Everything is all mixed up from the idea swap. Although some of the chromatids have stayed the same, some of them are walking away from this encounter quite changed. It's like returning home from a summer at camp. Maybe you've met a boy or girl. Maybe you've learned archery, and you're changed. In telophase 1, as in mitosis, the nuclear membrane reforms. Cytokinesis occurs and pinches the cell into two daughter cells. So now, the daughter cells are 1N, haploids. They have one set of chromosomes each, but each set is a random mixture of mom's info and dad's.
Okay, so we've gone from a diploid to a haploid cell, from 2N to 1N. Aren't we done?
Nope. Look, the chromosomes are still made up of 2 chromatids each. They still contain twice the DNA, so we gotta pull them apart.